first type of cardiac drugs we're going to talk about are antiarrhythmic drugs. In order to talk about them, we need to talk about what an arrhythmia in the heart is. We're going to go over the types of arrhythmias and then the antiarrhythmic drugs. So this is a normal ECG, and this is what's called a premature ventricular contraction, or PVC. We're going to talk about those. And initially, when you look at ECGs, you just want to be able to recognize what is abnormal from what is normal. So you always want to be on the lookout for that. We'll talk about the consequences of this particular pattern. It's really, one of them is really not a problem, but you need to be on the lookout for them, especially with an animal under anesthesia. So what is an arrhythmia? An arrhythmia is an abnormal electrical pattern of the heart, so that we're not getting the proper electrical conduction in the order that we would normally have it. It is originating not necessarily from the SA and AV nodes. It's originating from an area of the heart that we call an ectopic focus, or if we have a variety of them, we call them ectopic foci. And they are damaged myocardial cell or cells, and we're we'll talk about where they've come from, but they are the ones that, if you remember, each myocardial cell has the ability to depolarize in an automatic sort of way, an automaticity. So if its neighbor, and this is what the heart relies on, is a conduction from the SA to the AV node, and then along the bundle of His and all of the areas that transmit that signal. They rely on the cells uh, to stimulate the next cell, the neighboring cell. So if we have the stimulation originating from one of these ectopic focus areas, it's going to stimulate its neighbors to contract, okay, depolarize and contract. So that is not a normal rhythm, and that's not something that, in some cases, it can be life-threatening. So we want to make sure that we take care of it. So where these arrhythmias come from these ectopic foci, they're just damaged myocardial cells. And so what's going on with them is that sodium is leaking into the cell and causing a spontaneous depolarization. They're just more likely to depolarize on their own. And then they, again, stimulate their neighbors to fire. And the heart is then contracting in an abnormal fashion. So we can describe arrhythmias, and we typically think first of the heart rate that they induce. If they create a fast heart rate, we call that tachycardia. And if it is a slow heart rate, we call that bradycardia. You guys are hopefully familiar with those two terms. And then the next thing we do is we look for where that ectopic foci or lesion is in the heart, because that is where it's going to originate from, and then we can name it. So if you look at this pattern here, what we have going on is atrial fibrillation. We have these P and T waves are kind of on top of each other, and we've got sort of a normal ventricular rhythm going on here, but they're firing awful fast. So this would be a fast rate, and we call it a fibrillation because it is so fast, but it's a tachycardic type arrhythmia. And it's originating from the atria. So you want to kind of contrast that. So this is typically where we get the idea of where the ectopic foci is from looking at an ECG. And if you look at these, on the other hand, these are relatively normal, although, The P's and the T's are kind of mixed up in here. But this is a ventricular abnormal pattern. These waves are very large. And so that is originating from the ventricles. Sometimes it depends on where the leads are, whether they go below. Here's our normal, and here it's so large it's starting to go below the line, or whether they go above. And that has to do with these leads where we put them on the animal, and you're going to be more acquainted with that when you get to um, anesthesia. And that will cause the tracings, and basically those leads are on different parts of the body, and so the electrical activity is measured in a different way depending on which side of the heart, and whether it's cranial or caudal in the animal. But regardless, so those are large QRS waves, so that is definitely a ventricular arrhythmia. In this case, this is ventricular fibrillation, and the rhythm is irregularly irregular. You can't see that too well here, but we would normally measure. That's why we have these little boxes. We can measure the exact number of boxes between each thing. And if you look carefully, these are not equal between, the space is not equal between when the ventricles fire. And it's irregular. It's not forming a pattern. So irregularly irregular is ventricular fibrillation. You should just be kind of on the lookout for that. That's how we describe that one. Ventricular flutter, we have these little sawtooth patterns. So this is not a very effective beat of a heart. That's very shallow. What's going on there is those ventricles are hardly filling, and then they fire. And that is not compatible with life if it goes on very long, because we're simply not getting enough blood around to the body. If the ventricles aren't filling enough with blood, and they're just firing off and pushing little insignificant amounts around. So either of these is pretty big deal if you see that. So you just want to be, you may not recognize exactly what it is or where it's coming from, but you want to be able to point it out. But this is how we determine whether it's coming from the atria or the ventricles. So we may call it, I'm just going to go back here again. In this case, we call this atrial fibrillation. Um, that's the name of this arrhythmia, but it could also be called a supra, as in above, ventricular arrhythmia. So it's originating in the atria, supraventricular. So we'll sometimes call arrhythmias from the atria that. So this particular arrhythmia you need to be familiar with, a PVC. It's fairly common to see this in an animal under anesthesia that's hooked up to an ECG. One is okay, but if you start to see two or a series of them, you definitely, I would, even with the one, I would alert your veterinarian. But if you start to see a number of them, then we're going to be treating it. And it just indicates that this is premature. So normally we'd have the P wave, right? That's the atrial depolarization, followed by a little pause, the P and the R interval. And then QRS is the ventricles contracting and firing. So they have the atria have contracted and pushed the blood into the ventricles, little pause in there. Then they push the blood out together, the two ventricles. The QRS wave is what that's about. And then the repolarization of the ventricles is the T wave. In this case, we have no P wave. We have no atria filling here. This is simply the ventricles just depolarizing on their own for whatever cause. So that is what we call a wide bizarre wave. And you would want to alert your veterinarian at the first one, but if it's just one, we usually don't treat it. If we start to see more than one, then we're going to treat it. So that is when we become concerned. So when we're using antiarrhythmic drugs, we're trying to control the ectopic foci, or make that The electrical impulses go in the pattern that they should be going in, the normal pattern. And the goal is to create a normal rhythm in the heart. So not too fast, not too slow. We're going to talk about different classes of these antiarrhythmic drugs. So the first class, class 1, they inhibit sodium influx into the cell. So they're going to, if we have a cell that it's leaking the sodium in, and that's why it's become an ectopic foci, we're going to make that go slower. We're inhibiting that sodium going in. That will make the cell more stable and not depolarize on its own. We're going to talk about beta blockers, and that has to do with the beta 1 and beta 2 receptors that we talked about in the sympathetic nervous system. And then class 3, and these are two that we don't commonly use. There's a condition in boxers that we may use these for, but they block the potassium coming out of the cell. And again, help to make it more stable. But we don't tend to use these two. There's sotalol and amiodarone, and we'll talk more specifically about them, but they are not used so much as antiarrhythmic drugs. And then, at least in animals, in humans, they're used a bit more. And then class 4 are calcium channel blockers. Calcium, if you remember, and it was in that video that was in the first video question that I gave you, that calcium is what helps cells to depolarize. So whether it is a regular muscle cell or a cardiac muscle cell, we need calcium for that contraction of the cell. And if we block the calcium coming into the cell, that's going to decrease its ability to depolarize. So while we start it with sodium coming into the cell, the actual action is accomplished with calcium. So blocking it is going to help us get those cells firing back to normal. So we're going to, in the next lecture, we're going to start going through these different classes of antiarrhythmic drugs.